Good morning. The title of my presentation today is looking at current knowledge for immunological biomarkers in ME/CFS. At this point in time, I'd like to acknowledge Don Staines as well as Ekra Banu, one of my PhD students, for their contribution with the research I'm about to present. And there are other co-authors here, Daniel Peterson and some other PhD students of mine that have also contributed to the research I'm going to present here. In terms of the characteristics of ME/CFS, you would all know this. The exact pathomechanism is currently unknown. There is no diagnostic test at the moment for early diagnosis of CFS. Rather, it's a long, protracted elimination process for diagnosing ME/CFS. But what is characteristic of ME/CFS is there is immune dysfunction. And when you look at what studies have been conducted in terms of ME/CFS immunological investigations, I'll draw your attention to a number of specific cells located here on this figure. Most importantly, if we look here, neutrophil function's been studied by our group, and we've been looking at phagocytic activity as well as our oxidative burst capacity. That is how an actual pathogen is eliminated by this cell. And we've shown that it's significantly reduced, suggesting that there may be an innate associated dysfunction in ME/CFS. In addition, if you look at the literature that's been producing immunological investigations in ME/CFS, first and foremost, there's been a large body of information on natural killer cells. I'm going to focus on those at the start of this presentation because the majority of the literature shows a consistency from independent laboratory groups showing natural killer cell lysis is significantly reduced. Furthermore, if we look at what else has been investigated, our group and, to our limited knowledge, one other group has focused on specific T cells that are Cd8 cells. They are the sister to the natural killer cells, where they also produce a lysis factor to eliminate pathogens. The mechanism by which each of these lysis cells effectively destroy the pathogen is under two distinct different mechanisms, but the final outcome is actually lysis of that pathogen. Now, what else we have focused on, and Don started to talk about that in his presentation, and that is T regulatory functions, or Tregs as we call them. Our group most recently has focused on Tregs and potentially how Tregs may be implicated in ME/CFS. Now, there are illnesses that have Treg autoimmune disorders. So for that reason, our group has focused on this area, and I'll present some of that research later in this presentation. If we look at, finally, the B cell population, and my colleagues here in the front seat, Professors Flug and Mel, they most recently did a double-blind placebo-controlled study, and they used an intervention where they found that the anti-CD20 drug, rituximab, actually reduced the amount of B cells, suggesting that there may be an autoimmune disorder, or ME/CFS is an autoimmune disease. So the research that has been independently done by notable groups around the world has come to the conclusion that yes, there is immune dysfunction, but what are the actual main consistencies that have been shown in these immune dysfunctions with ME/CFS patients? And the next slide will give you that answer. And that is the consistent results that seem to appear at the present time from these independent research groups is most notably there's a reduction in NK cell numbers. Now, there are research groups that have reported no change, but if you look at the majority of the literature, it does suggest that there is a reduction in numbers. Coupled with that, and this is the most important component, there's a significant reduction here in NK cell lysis that is characteristic of ME/CFS patients. I'm going to talk to you about what may actually mediate that reduction in NK cell lysis later in this presentation. In addition, what also has been shown in the literature and has been reproduced by other groups is there's a reduction in an intracellular perforin protein. Now that's responsible for lysis in NK cell lysis and Cd8 cell lysis. So perforin is basically, if you think of a cylinder, it's a cylinder. It bores holes in the actual pathogen, and the cylinder is the perforin, and it changes then the osmotic pressure, and that causes then the cell to lyse. So there's been a reduction in also perforin, and I'm going to present some data in terms of the cell, the perforin, what the actual other proteins that may also play a role in this lysis, that is granzyme A and granzyme B, etc., how they also have been shown to be significantly reduced. So, in terms of what is emerging as immunological potential biomarkers for ME/CFS, this slide depicts what is actually being consistently shown by independent groups all around the world. And that is, this first study in our group, we showed there's a significant reduction in a particular phenotype of natural killer cells. I'll explain that phenotype in the coming slide. But nonetheless, the phenotype that is reduced is a characteristic now that seems to be assisting with showing early markers for ME/CFS. In addition, what mediates, this is the question, what mediates a reduction in NK cell lysis? We accept that it is a reduction, but what's the mechanism associated with that? And our group has investigated this in a very comprehensive manner, only to finish it this year, and it's suggested that it's actually one or two receptors on the natural killer cell. And these natural killer cell receptors, affectionately referred to as a Kerr, that's not Jonathan Kerr, who's a wonderful ME/CFS researcher, but a killer immunoglobulin receptor complex. There's numerous of those located on the natural killer cell. And we've investigated these Kerr receptors to see if one, if not more, has been responsible for these changes in NK cell lysis, and I'll present that data today. So I guess what I'm saying is, with the changes in phenotype that are being characterised now, as well as the potential for these Kerr receptors to further characterise NK cell lysis being reduced, we're building a further suite of characteristics that may be able to be used for immunological biomarkers for ME/CFS. In addition, if you look at the other cell I referred to, and that's the T cell that is playing a large part of lysis, that's known as the Cd8 cell. And as I said to you, it's the sister cell associated with natural killer cells. Now, there's been two groups that have looked at Cd8 cell lysis, and again, this is showing consistency that Cd8 cell lysis is reduced in ME/CFS patients as well. So again, we're building this nice repertoire of immunological markers that may now assist for hopefully expediting an early diagnosis of ME/CFS. In addition, if we look at the literature presented here, as well as the upper part of here, these are proteins or cells, but what controls those cells is actually at the gene level. So we've taken it further, and the research that we have done is we've looked at the gene expression of particular proteins. These proteins are the lytic proteins, the perforin, the granzyme A, that are responsible for lysis. So we've wanted to see whether or not at the gene level there is something controlling this reduction in NK cell lysis. Now, furthermore, the evolution of this component here in genomics has had a surge of actual recognition, and that is microRNA. And I'll talk about microRNA, but essentially microRNA control the regulation of gene expression and then ultimately protein synthesis. So I guess if you look at the emerging data that I'm going to present here today, we know characteristically that from the large amount of literature, we have a reduction in NK cell lysis. We have a reduction in granzyme A and to a lesser extent perforin. But these investigations here now are taking it further, and we're actually further characterising other immunological biomarkers potentially that can be used for early diagnosis of ME/CFS. So if we just take a brief overview of what constitutes the function of a natural killer cell, if we look here, the natural killer cell, we know it's responsible for lysis, and it does that through a number of, recognising a number of viruses or other foreign pathogens. And it does that through a perforin, granzyme A, granzyme B mediated mechanism. That's one phenotype of a natural killer cell. The other phenotype is a natural killer cell that activates other cytokines. It recruits other immunological cells to that site for assistance to resolve an actual pathogen. So the data that we have undertaken has to be, first and foremost, to look at that natural killer cell phenotype, those two phenotypes, they're called bright and dim. And in addition to that, we have then gone further and looked at what has been presented in the literature, and we have then decided to look at other different granzymes, granzyme Z, for example, and see whether or not that is also an additional immunological biomarker potentially for early diagnosis of ME/CFS. So if I just show you this diagram here, here's the natural killer cell. The mechanisms I was referring to as in what constitutes lysis is through these granzymes, these proteins, as well as I said to you before, this cylindrical pore that's called the perforin, and they function collectively to induce lysis of the target cell. So previous literature that has investigated NK cell lysis, there is a large body of that suggests that perforin, natural killer cell lysis are very good markers consistently for NK cell lysis. But we've looked at these granzymes and we have then characterised them further and shown there's a significant reduction in those as well. So I guess you have a brochure in your booklets today, like in a satchel, and it gives a historical perspective of immunological dysfunction or in ME/CFS. What you need to take home from that actual brochure is, first and foremost, natural killer cell is probably the most consistent at this point in time. A reduction in its lysis, as represented here, for immunological biomarkers for ME/CFS. So that's first and foremost one take-home message. The second take-home message is one needs to be sure that NK cell lysis is consistent over time. What you need to realise is when we do these immunological studies, the availability of people that will contribute to biomedical research is very hard to actually keep them engaged. And when you're doing work with ME/CFS patients, I understand how hard and how tiring it can be to actually be involved in something that's extra to your daily activities. So what people traditionally do when we're doing research, we take a snapshot, and the research that has been done on NK cell lysis has been at one point in time, not consistently monitoring NK cell lysis over time to ensure that NK cell lysis is significantly reduced and therefore is a reliable marker over time. And what we have done as the first to do this, I've got this data here, and the data shows NK cell lysis over a period of 12 months. Now, this data is for only 12 months, but there's more data coming out. We've done it out to 2 years. And this gives us confidence that the research that people have previously found with a reduction in NK cell lysis is going to be consistent over time. So when this sample is taken anywhere when we're doing ME/CFS work and we measure NK cell lysis, we can be assured that it's true irrespective of the time points. Similarly, what also we need to consider is the severity of ME/CFS. So this study, we investigated natural killer cell lysis, and we wanted to see whether or not a severe ME/CFS group, that is people that are bedbound, if they showed any changes in NK cell lysis compared to controls, if you can ever have a healthy control. And again, it was significantly reduced. But of interest here is the severe group with this natural killer cell reduction in their lytic activity. It was the same as the moderate group. So there seems to be disease severity does not necessarily correlate to the actual natural killer cell reduction. So in terms of further emerging immunological evidence for early biomarkers for ME/CFS, our group undertook a study, and we've consistently done this in other continuing studies, to look at the particular phenotypes of natural killer cells. And what we can see is there is a significant difference between these two populations of natural killer cells. These are called natural killer cell bright. Now, what this means is this particular phenotype may add further evidence now to build a suite for additional markers for early diagnosis, potentially for ME/CFS. And again, we looked at natural killer cell phenotypes over time, and again, it was consistent. It was significantly reduced over time between the healthy control group. And this data is only presented to 12 months, but again, it goes out to 2 years. So we know now we've got further evidence to actually maybe include phenotypes as another marker for assisting in diagnosing ME/CFS. So before I talk about the rest of my presentation, what I want to preface this start of this part of my presentation with is natural killer cells are regulated by T regulatory functions. Cd8 cells are regulated by Treg cells as well. In addition, how they regulate, that is, how Tregs regulate, we investigated potentially by looking at specific receptors on these natural killer cells. As I said to you at the start of the presentation, they're called KIRs, killer immunoglobulin receptors. Now, this diagram here outlines a number of KIRs, and we investigated whether or not lysis, a reduction in NK cell lysis, may be a result of a specific receptor on these natural killer cells that may be dysregulated or upregulated. And what we found is this particular receptor here, it's called a KIR3DL1, is significantly increased. Now, what's the importance of this receptor on this natural killer cell? Increased receptor expression of this natural killer cell is actually down-regulating. It's responsible for down-regulating natural killer cell lysis. So an increase in this actual KIR receptor is correlated with a reduction in NK cell lysis. So this is important. We think we're finding out where a potential mechanism may be for explaining how we see a natural killer cell lysis reduction. In addition, as I said to you a moment ago, Cd8 cells, there's an emerging component of ME/CFS research that's looking at Cd8 cells. And currently, this information suggests that Cd8 cell lysis may also assist in building early or immunological biomarkers for ME/CFS. And again, we measured it over time and we found a significant reduction between the control group. So we've got confidence here that we can now potentially add Cd8 cells and the measure of Cd8 lysis with NK cell lysis to build this repertoire of immunological biomarkers. Now, in terms of cell function, you look at natural killer cell lysis, you look at Cd8 cell lysis, and you say, what mediates that? Well, it's at the gene level, and it's through perforin, it's through those granzymes I was speaking about at the start of this presentation. So we looked at the gene expression of perforin, and we also looked at the gene expression of a number of other proteins, GZMA, GZMK, etc., for how there may be changes in these proteins, which then affect, obviously, NK cell lysis. And what you can see is there's a significant reduction in GZMA. So what this suggests is now we can use, potentially, the gene expression of GZMA to characterise another biomarker, an immunological biomarker, for early diagnosis of ME/CFS. So we're building not just NK cell lysis as the be-all and end-all. We're building this repertoire, and I think that's very important to realise, because we can't just solely rely on one marker. I do not think that will ever be how it will be. In terms of this component of research I'm going to present, I touched on it at the start of this presentation, and I said we've looked at the cells, we've looked at then the gene component of what makes those proteins or those cells. But what actually controls those genes are these actual proteins, and they're called microRNA. Now, they're small, very small, non-coding proteins of RNA, and they're very small in terms of their nucleotide length. And what they do is, this is the important part here, they affect the actual translation of these genes at the post-transcriptional level. And I'll explain that in the next slide. And how they actually do it is they bind onto the messenger RNA and they facilitate either degradation or a stopping of that transcription of that protein. So why do they do it, and how do they achieve it? They actually achieve it because they're complementary to the messenger, to the actual messenger RNA that's being transcribed to make the protein. So if you take a look at this diagram, this is a very small snapshot of what microRNA do. MicroRNA are obviously generated in the nucleus, they're transcribed by these two proteins, Pasha and Dosha. They're not named after dogs, but they sound like they are. In terms of where they occur, they form this hairpin pre-microRNA structure. They're exported through this protein called exportin 5. Consequently, then this protein in the cytoplasm, referred to as dicer, chops it up and makes it a single-stranded RNA molecule. Consequently, when it combines with this protein here, RISC as it's called, it ensures that it's a mature microRNA. Now, the take-home message for people today is this microRNA single strand binds on to the messenger RNA here, as represented in 1. And what it does, it only allows partial transcription of that gene. So it affects the actual protein, if at all, that's going to be produced. In addition, microRNA, the mature microRNA, is able to then ensure degradation of this messenger RNA. Hence, there's no transcription of that protein. So what we have done in our lab is to look at specific microRNA that control natural killer cells as well as control Cd8 cells, because the literature shows that they are potentially where you're going to get consistency. So, of course, it makes sense that then we would investigate those microRNA. And from the results, what we did is we specifically honed in on 19 particular microRNA that are specific to natural killer cells and Cd8 cells. Now, the 19 microRNA are responsible for cell apoptosis, for cell cycling, cell development, and regulating the immune function of Cd8 and natural killer cells. Now, the research that I'm presenting here today characterises specific microRNA that of those 19 microRNA, 9 was showed to be significantly different. That is, significantly decreased in the ME/CFS patient group that we studied. I'll draw your attention to these bars here. These microRNA 106, microRNA 21, 150 were shown in natural killer cells to be significantly decreased. Now, the importance of microRNA 21 is it's shown in the literature to regulate Treg cells, and Treg cells have been implicated in autoimmune. That is, increased T regulatory function has been, in some illnesses, suggested to be a key factor in autoimmune disease. In addition, if you look at the Cd8 cells, microRNA 21 again features. So we have 2 cells that are dependent with microRNA being reduced. So if we have a reduction in that, that may then essentially cause a change in T regulatory function. That is, Tregs may be increased because microRNA are significantly reduced and there's no controlling mechanism for those Treg cells. In addition, when we look at the natural killer cell Cd8 microRNA proliferation, we see then there's a significant difference again in this microRNA here, microRNA 146A. Now, the importance of microRNA 146A is it also is controlling T regulatory function. In the literature, it has been shown to significantly play a key role in regulating Tregs. And Tregs, as I have said, are suggested to play a role in autoimmune disease. So at the gene level, we have looked at what's controlling these genes. And if you think about the gene for coding the Tregs, as Don was explaining before, we have a reduction now in these 2 particular microRNA that are responsible for suppressing T regulatory function. And because they are reduced, we would then think, and our other studies then confirmed it, that there would be a heightened increase in T regulatory cells. And how would we look at measuring T regulatory cells? There are a plethora of T regulatory cells. We characterise the T regulatory cells through a number of proteins that sit on the cell surface. Namely, we characterised it by a Cd4, as that is a T cell. We characterised it by a Cd25 protein. Furthermore, then we characterised it by a Cd39, as well as this component here, which is known as FOXP3. Now, FOXP3 is very important, and I'll explain in a moment, but essentially it is part of the Treg function that then also initiates, as Don was suggesting a moment ago, it causes the hydrolysis of ATP back to adenosine. And of course, how does that occur? Through that cyclic AMP mechanism Don was referring to. Now, where does that occur? On these T regulatory cells by this marker, another protein known as Cd39. So we characterised T regulatory cells. Now, as I said to you, I'll preface the explaining these results by stating, first and foremost, there are a plethora of T regulatory cells. We have characterised Treg cells collectively. And what we have shown is there is a significant increase in Tregs over a period of time in ME/CFS patients. How does that relate to the research that I have presented? First and foremost, if you think about the microRNA data, there's microRNA 146A, microRNA 21. They were significantly reduced. Because they're reduced, they don't exert their control potentially over these Treg cells, therefore suggesting that they may be increased. And Tregs in some illnesses have been characterised playing an important role in autoimmune disease. In addition, if you think about what Don was saying about the changes in purinergic signaling. Well, they're mediated, obviously, with those PACAP, VPAC1, VPAC2 receptors through the CD39 mechanism I explained to you. And we wanted to see whether or not there was a change in that purigenic signaling, I should say, and correlating that with the FOXP3. What we can see is there's a significant increase in both, suggesting a perturbation in this ATP mechanism with these T regulatory cells. So I guess where would this potentially be quantified? And Don showed you some receptors, PACAP, VPAC1, VPAC2. So we examined those 3 receptors to see whether or not this purigenic perturbation may be affecting one of these receptors on these lymphocytes we analysed. And what you can see is there's a significant increase in this particular receptor. Now, what does that actually mean? VPAC1 is quite ubiquitously expressed, and my colleague, Professor Delgado, will explain later after our morning tea the impact of VPAC1, VPAC2 and VIP. But essentially, if you've got this purigenic signalling perturbation, it's then classified by showing an increase in this receptor expression. So there might be a change then in this pathogenesis, with it being a cyclic AMP change with these cells. In terms of looking at what has been most recently reported in the literature, one would say that the results that was produced by Professors Flug and Mel changed the course of ME/CFS, I would say, last year, for a good thing. In terms of what may come, the title of my presentation is Emerging Immunological Markers for ME/CFS. I would say that this space is to be watched, and there may be further results that may provide a rationale for why we're thinking ME/CFS may be, I say this, may be an autoimmune disorder. So in summary, I would say currently there is very good evidence to say we have natural killer cell lysis as a characteristic of showing immunological markers for ME/CFS. I think you would know that. In terms of other characteristics that show ME/CFS as an immune dysfunction, we can gain confidence with natural killer cell phenotypes as well as specific proteins, that is perforin and GZMA, the most predominant proteins that show consistency being reduced in ME/CFS patients, both for NK cells as well as Cd8 cells. In terms of emerging areas that provide evidence for emerging immunological dysfunction in ME/CFS, potentially for biomarker usage, one would then focus on Cd8 cell lysis, importantly microRNA. Now, we only examine 19 microRNA. Currently, we are doing a whole microRNA study. There are approximately 1,500 microRNA that are responsible for controlling transcription at the genomic level. We are undertaking that investigation at the moment and should finish it by the end of June. In addition, the evidence that I provided today was a pilot study. We're currently doing further studies to look at subtypes of these Treg cells to see if it is a specific subtype of these regulatory T cells that is significantly upregulated. And I hope that with these investigations, this will provide further development for including these as immunological biomarkers for ME/CFS. Now, finally, I'll finish by saying the research that we've done has been in the last 4 years. It's been successfully achieved at Bond University. However, to be true to the patient cohort in Australia, I decided, in consultation with the group, I think we should relocate, and we relocated to the new University Hospital, a $2 billion facility, 15 minutes up the road from where we previously were at Bond, to interface with clinicians, to have places within the hospital designated for beds for ME/CFS patients, to monitor them continuously, and to provide an actual nexus between research as well as the patient-clinician cohorts. I'd like to acknowledge these governing bodies for funding, BD, the Mason Foundation over 4 years, the Queensland Government, and the Alison Hunter Memorial Foundation. Thank you.
Thanks, Sonya. Right, in the run-up to coffee, tea, whatever now, we have time for some questions. So there must be something rumbling in your heads, and so get stuck in. Never think a question is too simplistic and stupid. Just be stupid and ask the question. It's amazing how often you get a funny answer. Sir, and then it's you.
Hello, Dr. Gunnar Sandberg from Sweden. I have treated 2,000 TWAR, that is chlamydia pneumonia patients, and in the expression of clinical state, they are extremely similar to ME patients. I have also had some ME patients coming up from their wheelchairs and things like that. There is one very dominant trait, that is effusion of fluid from lungs and from ear, nose, throat area. And I have a strong suggestion, actually evidence of diminished oxygen pressure in the blood. I have 2 15-year-old boys now. One have 91% in blood gas analysis of oxygen, and he is lying down, and the other one is 94. He can be up, but it's not participating in any sort of physical exercise. Now, have anybody seen the influence of this primary finding with the diminished oxygen input in the blood?
Right. Sonya or Don, did you know?
On these systems?
Yes.
I can't speak specifically to your question about have patients been identified with either oxygen deprivation or the fluid imbalance that you imply, but just on a theoretical basis, I suspect there is some association with aquaporin receptors in this condition. You might recall aquaporin receptors seem to have a role in neuromyelitis optica, NMO, and it maybe has some autoimmune role in that condition. However, the aquaporin receptors are certainly very much implicated in fluid balance, fluid retention, and so on. So perhaps that would be one area of research. The second, you mentioned a relative hypoxia, and I mentioned that in my talk, and I think this is a very important point, that patients with ME/CFS may well be hypoxic at the intracellular level, if we can put it in those terms, because we mentioned oxygen-glucose deprivation, OGD, and I suspect that is very much integrated with cyclic AMP functions of another type. So all I could suggest is to go to the literature. I would suggest you look at aquaporin. I would suggest you look at OGD, oxygen-glucose deprivation, and that may have some bearing. But apart from that, we don't have enough clinical experience to answer the question directly. I'm sorry.
Is that okay, Sonya? That's fine.
Yeah, that's fine. I don't know of any knowledge of that.
No, just pointing out that there is actually some good data out of Stacey Stevens' group, as well as actually the Lights group in Utah, documenting that there's low VO2 max at baseline, as well as with exercise in many, actually the majority of ME/CFS patients. So to the second part of your question, I think there is good evidence for that.
Okay, let's have another couple of questions. One here, please. Yes.
I have ME, can be seen. I'm also principal carer for 2 people with very severe ME. Now, I've got 2 questions. First is, you're talking about biomarkers. Are we then looking possibly to a blood test, a saliva test in the future that will show these biomarkers? Okay. The second question is related to what's just been asked. With the hypoxia, we're finding with very severe that oxygenated water can improve cognition, combined with the taking of the organic version of coconut water which is also being used by athletes when they're exercising. So you were talking about the glucose need and the oxygen. This seems to improve cognition quite fundamentally. So how do all these things link together? So the biomarker first, and the other one second, please.
Okay, I think first and foremost, if you look at biomarkers, you have to have consistency over time, and they have to be reliable, and we have to test them in other population groups to show that they are distinguishable and unique to a particular population, that is, in this instance, ME/CFS. So I know there's overlapping symptomologies with, say, for example, MS, and for that reason, I think that we need to test whether or not these are unique in ME/CFS. Otherwise, we could muddy the waters. Currently, we're undertaking such a study with Queensland Health as well as with Dr. Peterson, and it's a national study. So I think watch that, because it'll either show promise, or I think a negative result is also an outcome that is not deleterious. I think it's a positive outcome because we're not barking up the wrong tree, so to speak.
Information.
Exactly.
The more information we have, the better
Yep.
we can go forward.
Exactly.
And in respect of the second question, I'm afraid I can't be drawn on that. It may amount to a testimonial for coconut oil or something, so no, I won't. I can't comment. Yeah. Well, I look forward to seeing published data on that.
Okay, one last question. Yes, please. Thank you.
This is an answer about the question that's been asked. The work in hemorheology by Dr. L.O. Simpson indicates very clearly that the red blood cells population changes shape in many chronic illnesses, but particularly in ME/CFS. Micrographs of immediately fixed samples of blood will clearly show the non-deformable red blood cells, and this has a fundamental effect in the delivery of oxygen through the microcirculation. This work has been known for years, and it's been ignored for years. But many people will remember Dr. Simpson because he attended many meetings and in the 90s. Hemorheology is absolutely ignored for some reason.
Can I answer that question? In 2010, I collaborate with Herbert Meiselman and Olgs Bazkert, and they are the world leaders in hemorheology. And I undertook a study on 20 ME/CFS patients and looked at the biorheology of ME/CFS patients compared to controls. That was published in Journal of Translational Medicine in 2010. And what we investigated was first and foremost red blood cell aggregation, because if there's, at the microcirculatory level, if you're going to look at potentially how fatigue may develop, it may be through the red blood cell, and there would be presumably A aggregates, and that would then impede the exchange, that microcirculatory exchange, as well as then looked at deformability, that is, how well the cells deform. Now, from that study, and that was very critically reviewed, and I can say that there was no significant difference in red blood cell aggregation or red blood cell deformability in ME/CFS patients. So I think the microcirculatory component is only one component of the actual illness itself. What mediates that, I think, is more of a central component. It might be a neuroendocrine mechanism. So, and then there was another independent study we did on that red blood cell aggregation and deformability, and we looked at protein cell surface markers, and there was no significant difference again. So at the cell level and at the protein level, from those two studies, which are the first two to do that in ME/CFS patients, there was no difference.
Okay, thank you very much.